Hi guys, welcome. Sorry, it took me a second to get it all going. I'm hoping for those of you who are with me on TikTok that you are able to see my screen. And for those of you who are with me on YouTube, I hope you can still see my screen as well. And I hope we're all doing well. Chidima, how are you? Are you feeling better today? Hi guys, if you're joining me, welcome. If there's anything you would like to cover, please do ask away. Alex, how are you? Daryl, how are we doing? Sorry, I'm just getting up so I can see what everybody is doing. How are we? Is there anything specific we want to cover tonight, guys? Please do ask away. And I am free to do whatever you want to do. Right, here we go. Oh, Chidama, I'm sorry to hear that. Also, please help. I need a photosynthesis and respiration test tomorrow. May we please go through it? You need to cover photosynthesis. Literally forgotten everything. Of course we can do that. I should probably take time out. You proceed with the biology though. Oh, Chidama, hope you're okay. Hi! Do you do A-level biology? Yes, I do. And Alex has requested us to do some photosynthesis. Chidima, can you do me a favour? Can you send me an email to ukbiotutor@gmail.com with your email address, and I will respond to you via email. So I want to send you something. Don't polymerases go in a 5? So polymerase can only build in a 5-3 direction. So make sure you're talking about building in a 5-3 direction rather than saying that it is going to be moving in a 5-3 direction. So be careful on that one. But 2, one strand acts as a template strand. RNA polymerase binds and moves along this strand in the 3 to 5 direction within the base sequence. So again, be careful that Darrow, I missed one of your messages that you said before there. Quickly explain 5-3 prime direction and 3-5. I don't get which way around it is. Yeah, it's all about the building in the 5-3 direction. What should you email? Just send me an email saying, hi Chidima, that's all I want you to do. So I have your email address and I will then respond to you with, I want to send you something that hopefully will help on that one. So send me an email so that I can then respond to you via email. Right, Alex, I've not forgotten photosynthesis. Alex, where are you struggling with the most? Light dependent reaction or light independent reaction? Which of the points is... Yeah, both of them. So Darrow, to answer your question, both transcription and translation, whether we're talking about DNA polymerase or whether we're talking about RNA polymerase, both of them build in a 5-3 direction. It's the only way they can build. And the reason being is because the third carbon is the place where there is the hydroxyl group to build the phosphodiester bond with the phosphate group. So it's important that you look at that part. Right, let's start with the light dependent reaction then and move to the light independent reaction for that one. Okay, so hi Tilde, welcome. Could we do oxyhemoglobin? Study talk, if you are looking specifically for oxyhemoglobin, then what I would suggest is go and double check my transport in animals videos, because I've got one on the Bohr shift and the transport of oxygen, and that would be good for you. I'm going to start with photosynthesis. Alex is a long-time serving student of mine on here, and so I'm going to start with the light dependent reaction, and then I'm going to move on to the light dependent reaction. So right, here we go. Let's bring in some content. If you are year 12, guys, this is one of the things I would say to you. You have got so much coming up. Sometimes it's a good thing to get ahead, sometimes not. But I would definitely say that watching the videos that I do will still help you on that one. But please do ask away, and I will answer questions as I go as much as I can. Okay, so as we are moving through this then, and as we're looking at photosynthesis, we're going to start with a light dependent reaction. One of the things for you to note, really important, is all about your photosystem. So when you do the light dependent reaction, you should know about the thylakoid membrane, and you should know that in the thylakoid membrane there are photosystems, there are proton pumps, there are ATP synthase. So we want to talk about what's happening, how it's happening, and why it's needed for the next stage. So when you look at the photosystems, they're made up of these light harvesting systems and reaction centers. So basically, you've got these molecules, xanthophylls, carotenoids, chlorophyll B, chlorophyll A. So you've got these pigments in the proteins, and what that means is that these pigments are going to be able to absorb light energy, and they absorb light energy at different wavelengths of light. Okay, so it's important to note that when they're absorbing the light energy, they transfer the energy that they're absorbing to the reaction center. So when we talk about the photosystem, we're talking about both the light harvesting system, so all the pigments, along with the reaction center. So the key thing to note is the reaction center only has chlorophyll A. So the chlorophyll A, which is located in that reaction center, is the one which can donate its electrons. So this is the important part when we're talking about the absorption of light and the electron transport chain. The electrons are only donated from the reaction centers of photosystem 2 and photosystem 1. So it's important to note when the photon of light comes in, the electrons get excited because they've absorbed that energy. They become excited, they jump up to a higher energy level. So when they're at a higher energy level, what that then means is that they're in an excited state. They've got more energy than they started with. So when they move back down to their original position, that excess energy is going to be released. So that's important to note. When they are absorbing that photon of light, they're absorbing the energy, they get excited, they jump up. When they move back down, they lose the energy. So the other thing to note, sorry, I need to move things around because I've literally just jumped this straight across. Organic cofactors, coenzymes. What have we got? We've got NADP. Now, I call it NADP for photosynthesis. That's not what it is. It is NADP because it's got a phosphate. So you know that NAD, so why they move back down the energy level. So the reason they move back down the energy level is because they've released the energy. We haven't spoke about how they've released the energy yet. All we spoke about is the fact that they can get excited, and when they do release the energy, they move back down to their original state. So when we talk about the electron transport chain, the electron transport chain is going to be accepting the electrons, transferring the electrons because it's an electron transport chain, and as it's transferring the electrons through the electron transport chain, it therefore brings it back down, and that releasing of the energy, Darrow's absolutely right, is going to make it to where it's at a more stable state. So it's important to note that the NADP is a coenzyme, and that coenzyme can accept electrons. Okay, so the NADP is the one for photosynthesis, NAD is the one for respiration. So I use the P to remind me that that is in photosynthesis. If I'm doing respiration, I would just ignore the P and put NAD. They are both coenzymes. They're both got the same structure. However, one has a phosphate, NADP, and one doesn't. Yeah, this is where, Darrow, it's really important that you note that your chemistry will come into biology and it helps you. Yes, P for phosphate, but I remember, yeah, that's exactly how I remember it because it's helpful. When the NADP accepts the electrons and hydrogen ions, what it then becomes is reduced NADP, and it can be oxidized. It can lose those hydrogen and electrons. So I again like to think of this a bit like a taxi. A taxi is going to pick up its passengers and then drop its passengers off wherever they are needed. So if we then look at the non-cyclic photophosphorylation, so before we move through anything, split the word up. Non-cyclic is not a cycle. It's going to go in one direction and off. Photo, to do with light. Phosphorylation, adding of a phosphate. So the minute you have these big, huge words in biology, excuse me one second, what you need to do is understand that you can split it up to work out what it's talking about. So whenever I do any of this, I'm always going to have photo, meaning light. I'm going to have phosphorylation, adding a phosphate to produce ATP. And because I'm saying it's non-cyclic at the moment, it means it's not going in a cycle. So off it goes. So what you have to know is that photon of light is going to come in. Now I'm going to start at photosystem 1. However, it happens at both of them at the same time, okay, simultaneously. So photosystem 1 is going to absorb the light energy. So the accessory pigments, the primary pigment, these are all going to be absorbing different wavelengths of light. We know that visible light has different wavelengths. If we go from the red wavelength all the way down to the violet wavelength, we know that they've got different wavelengths. Therefore, they've got different amounts of energy that is going to be absorbed. Is the light energy the energy released when the electron goes back to its original level? So no, it's not light energy that's released. Light energy is absorbed. The energy that's being released as it goes back down to its original state is the energy needed to actually pump hydrogens against a concentration gradient. So it's a kinetic energy. So the light energy is being absorbed. That light energy has then excited the electrons up to a higher energy state, and then as they are moving through, they will release energy by pumping hydrogen ions against the concentration gradient. So what we have is, if I go back a second, the electron, the photon of light has been absorbed. The electrons were donated at the reaction center, and they're donated to the electron transport chain. Now, these photosystem 1 electrons will make their way up through the electron transport chain to this part here. Now, you don't need to know the name of this. However, what you do need to understand is this is where my NADP is going to come in and become reduced. So when my NADP, my coenzyme, comes to the last part here, it becomes an electron acceptor. So the NADP has accepted the electrons and also the hydrogen ions. Starboy, this is for OCR, but it's also linked to AQA because you have to know the same content, although you don't need to know the names, non-cyclic and cyclic photophosphorylation, whereas you do on the OCR side of things. Hydrolysis, biological word, yes. Alex, I'm just quickly looking at what you said. Shown on the graphs. Alex, who are you answering? Hold on, is this only showing me the top chat and not all the chat? Have I missed something? I don't know how much I need to know for that. What have I missed? An electrophile. Alex, I'm getting really confused as to what you said in the chat there. I'm speaking while you're going through. Oh, who are you speaking to? Oh, I'm so confused on that one. Anyway, I'm going to go back to this and ignore whatever you're chatting. How similar OCR and AQA? They're very similar. Oh, you're chatting to me? Yeah, but you've said an electrophile, so I'm confused as to what you meant by that. Do you mean the electron acceptor rather than... That's what I'm confused at, why you said an electrophile. The light-dependent reaction occurs in the thylakoid. Yes, in the thylakoid membrane. I'm live at 9 o'clock on a Wednesday, for sure. Sometimes on a Monday. So I'm making a link to chemistry. Go away with your chemistry. We're not doing revision of chemistry right now. He's just calling in as you're going along. He's not replying to anyone. Awesome. Dara, I love that. I just got confused because I was like, hold on a minute. I missed a bit. Alex, no, it's not a problem. I just got confused because you said the electrophile. Anyway, back to this. We have the reduced NADP that's going to go off to the Calvin cycle. So we have our photon of light that's come in. The energy has been passed down to the reaction center. The electrons were donated. They have been accepted by the reduced the NADP to become reduced NADP. We have the photon of light come into photosystem 2. That is then going to make its way down to the reaction center. The electrons will get donated from the reaction center. They'll then make their way across from photosystem 2 across a proton pump here into photosystem 1. Now the key thing to remember, okay, and I say this in my lessons when I do this with my one-to-ones, if I have a pen in my hand, can I accept another pen? No, I can't, okay? I need to have lost that pen to then accept, this is where I can't find a pen. This isn't a pen, it's a paintbrush, but I'm going to take it anyway. So I can then accept if I have nothing in my hand, okay? So if I donate my pen to somewhere else, I need a pen back to replace the pen that I lost. So if I have donated my electrons from photosystem 2 and they have now been accepted by photosystem 1, because the electrons that were donated by photosystem 1 are then being accepted by the NADP, they therefore need to be replaced. Absolutely. Donated, the pen has gone, it must be replaced. So the electrons that were donated need to be replaced, otherwise this cannot continue. So we saw that photosystem 2 donated its electrons to the electron transport chain, and that therefore went all the way to photosystem 1. The electrons from photosystem 1 have gone to the reduced NADP. Now, by pumping those hydrogen ions against the concentration gradient, so just to note that this was a low concentration, this was a high concentration. We're in the stroma outside, and inside we're in the thylakoid space. So it's important to know where you are where this is occurring. Why do we want all the hydrogens in the thylakoid space? Where are the electrons donated to? Okay, so to answer Til's first question, why do we want all the hydrogens, the hydrogen ions? This should actually show, by the way, this is where I'm like, oh, I need to adapt my diagram. This needs to show hydrogen ions, not just hydrogens. These are hydrogen ions. And what we want is for them to move into, let me go back a second. So when they've gone against the concentration gradient, because the electrons moved from photosystem 2 to photosystem 1, the energy was used to pump the hydrogens against the concentration gradient. So what I have got here, if I move over here, is a high concentration and a low concentration. And because I've got a high concentration and a low concentration, they are going to diffuse down the electrochemical gradients. They're going to make their way out through the ATP synthase. And when they make their way out through the ATP synthase, what's going to happen is that motive force, the movement of them through this stocked particle, ATP synthase, is going to provide the necessary energy for the ADP and the inorganic phosphate to come together to form ATP. So we need energy to form that final bond to make ATP, and that is why this is called phosphorylation, because we are going to be adding a phosphate to produce ATP. So we needed the light energy to create a proton gradient so that they would then diffuse through. Okay, so Alex, no, we don't say it's facilitated diffusion, and this is one of the things you have to be very careful of. We don't say it's facilitated diffusion because what we're actually doing is we are doing what's known as chemiosmosis. So you learn about chemiosmosis when you did respiration, where at the Christie, oxidative phosphorylation occurred. We had the electron transport chain. We had a proton gradient. That proton gradient, then as the protons made their way through, that flow of those protons through the ATP synthase, that provided the energy for adenosine diphosphate and inorganic phosphate to come together to produce ATP. So we know that this is non-cyclic photophosphorylation because we have got that the electrons moved from photosystem 2 to photosystem 1. The electrons moved up and were accepted by reduced NADP, and they are going to go to the Calvin cycle, and our ATP will also go to the Calvin cycle. So the two products of the light dependent reaction that are needed in the light independent reaction are reduced NADP and ATP. Electrons get accepted by final electron acceptor, which is oxygen. Yes, if we are talking about respiration, then the oxygen is the final electron acceptor if we're talking about oxygen. However, in this case, we're not talking about oxygen because we're not doing respiration. We're talking about the light dependent reaction. So in this case, for the light dependent reaction, the NADP is the last electron acceptor of this electron transport chain. Good, Alex, I'm glad this is making sense for you. Ozzy, you are very welcome. I hope this is helping. Okay, so back to this then. What have we got? Well, our electrons that went from photosystem 2 to photosystem 1 need to be replaced. So the only way they can be replaced is if we carry out photolysis. So lysis to split, photo because of light. Because of the light energy that was absorbed by photosystem 2, what we have got is those electrons that were donated need to be replaced. So if we take water and we split it up into electrons, hydrogen ions and oxygen, that's then meaning that those electrons can be accepted into photosystem 2 to replace the electrons that were lost. Do we need to know about PMF? Sorry, abbreviations for me. What does PMF mean? When you're saying the PMF for movement for hydrogens. Yes, proton motive force. Yes, sorry, Azia, it took me a while to work that out. I think that's what you were meaning by PMF. You want to be using, yes, photosynthesis, year 2, proton motive force. Yes, So yes, you want to try, if you're aiming for that A star, then yes, you want to say things like the proton motive force. If you're not too worried about trying to give those definitions, describe what is happening. So if you just describe it, the hydrogen ions are flowing through the ATP synthase from a high concentration to a lower concentration, providing the necessary energy to join the ATP molecule. That will be enough to get you the marking point. So if you are not sure on things like the proton motive force, then describe what is happening, because that will make a big difference. Okay, so that is the non-cyclic photophosphorylation, where we have the electrons moving through, and the reduced NADP and ATP are going to go to the Calvin cycle. And if we were looking at the cyclic photophosphorylation, one second, let me move that title across. I think this stuff is easier than anything else that's plant related or gene sequencing modules. Oh, Azzy, all the plant stuff I've put together, the only thing that's missing is plant defenses. I haven't finished plant defenses yet. And the gene sequencing modules, I'm actually currently making my study guides for that. So if you haven't checked out my study guides, please do check out my website, ukbiotutor.com, where I have put the study guides on there of the ones I've done so far. I'm currently working on patterns and inheritance, and I'm also going to add in the manipulating genome when I get a moment. But obviously I have to factor in where my money can come in, and also I like to do these sessions for you, because sometimes asking the questions is what gets that understanding as we go. Okay, so back to this then. We have got cyclic photophosphorylation. Oh, Daryl, thank you. I'm glad you like them, and thank you for buying them all. I do really appreciate that. It is, I think I'm probably going to give it one more week at the discounted price, and then I'm going to hike them back up to the original price. But yeah, thank you. I do appreciate it. Cyclic photophosphorylation, slightly different. It's cyclic this time, but still the same photophosphorylation. So we've still got the photon of light coming in. We've still got the excited electrons, but this time, oh, what happened there? There was no reduced NADP. That's a lie. There was no oxidized NAD. So remember, on our last one, we had got an oxidized NADP coming in, and that would then accept the electrons. Well, there isn't a oxidized NADP, so I'm going to get rid of that. In fact, no, I won't. I'll put it back in there. But I'm going to note on here that is not available. Remember I said before that the NADP is a bit like a taxi. It picks up its passengers and off it goes and drops them off. Well, if you have a taxi that's not able to drop off its passengers, i.e. my reduced NADP is still up there, because it can't drop off its passengers, it cannot come back to pick up more passengers. So there is a delay. And if there's a delay, what that therefore means is those electrons, if I go back, that photon of light that's been absorbed by photosystem 1, the electrons can make their way, the hydrogens go against the concentration gradient, therefore we can still produce ATP, so we can still do photophosphorylation because of light, the light coming in, the light being absorbed, light energy, those wavelengths of light being absorbed, the electrons get excited, but they're not able to be accepted apart from back from where they're donated. So they're still going from the electron transport chain, but they're going back to photosystem 1. That is cyclic photophosphorylation, because we're adding a phosphate. We're adding the phosphate to the ADP to produce ATP. We have done this because of light. So it's really important you know the difference between the light dependent reaction and the light independent reaction based on the products that have been made. So if we quickly go through the light independent reaction, then that hopefully will help you, Alex, for a bit of your test that you've got. Okay, so non-cyclic photophosphorylation, cyclic photophosphorylation. What have we got? The light independent reaction. So it's known as the Calvin cycle. It doesn't require light. It takes place in the stroma, and it's going to use the products. So if this is my thylakoid, each one of these is a thylakoid, therefore I have a granum, because I'm looking at the whole thing. I don't know if I can write upside down on this. Let me just try. Granum. Wow, that's really hard to do. My writing's bad as it is, but then try to write sideways really doesn't do. Do you tutor IB biology as well? Yes, I do. What was I writing here? Granum. I love IB. I miss, to be honest, I do miss teaching the IB, actual teaching it, because I love it so much. I love the way it just flows. It just all is amazing. Anyway, back to this. The granum is the stack of thylakoids. The thylakoid is obviously where we have our electron transport chain and our ATP synthase. Therefore, what we understand is that with light coming in, water for photolysis, it produces oxygen as a waste product. We are going to produce ATP and reduced NADP, and they are going to go to the Calvin cycle, and they're going to be used to fix carbon into the molecules that's needed. So the exam board that I'm doing right now is OCR A. However, I'll try where I can adding in bits which are AQA. So if you're AQA, you have to know about the excited electrons, calling it photoionization. So that's the extra little bit that you need to know for yours. You don't need to know cyclic and non-cyclic photophosphorylation as the name, although your teacher may have taught it to you. Hi guys, Rowan, how are you? How are things? Thank you for joining me, guys, and let's keep going. So we're currently at the light independent reaction. We made reduced NADP and ATP in the light dependent reaction. So now we're going to use them. So we have this RUBP. So the RUBP is, that you need to know its name, is ribulose biphosphate. So we have a 5-carbon molecule. This 5-carbon molecule has 2 phosphates attached, which is why it's called RUBP. Now, carbon dioxide. Respected teacher, I apologize for reaching out unannounced. May I respectfully inquire whether you provide AP Sasha, I'm really sorry. I don't do chemistry classes because I haven't actually done the content or know it myself to that level. I do know someone who does OCR chemistry tutoring that I could advise you on, but with you saying advanced, I'm not sure if they would be able to offer that one. But if it's OCR, then I do know someone who does tutoring if you're looking for that. So send me an email or a message and I will respond with their email address. Yes, good. Oh, Rowan, I'm really glad to hear that you're doing well. I am well, thank you. Even though I am cold but warm at the same time. It's a bit of a strange one. Right, ribulose biphosphate. We have a 5-carbon molecule. What's going to happen is carbon dioxide and the ribulose biphosphate are going to be combined together through the use of an enzyme called Rubisco. So this is important to note that we have got Rubisco, which is our enzyme ribulose biphosphate carboxylase. So that is an enzyme that is going to catalyze the reaction, and it's going to catalyze the reaction at a fast rate because we're talking about an enzyme. Okay, so that's the way to remember it. Now, when we have a 6 carbon molecule, so right now we have a 6 carbon molecule, that 6 carbon molecule is unstable. It does not stay as a 6 carbon molecule. It is going to split apart and it is going to be converted into 2 3 carbon molecules called glycerate 3-phosphate. So we've got glycerate 3-phosphates. Now, be careful. You need to know the name glycerate 3-phosphate. You need to know the name ribulose biphosphate. Do not drop them down to GP and RUBP until you've written it at least once. Pepsi Max, do I need to know the other reaction that Rubisco catalyzes? Yes, you do need to know about photorespiration as a response to the limiting factors of photosynthesis. So yes, you would need to know about that. However, they don't want you to go into loads of detail. If they're going to ask a question on that, they'll be giving you some information, and it'll be more looking at you understanding that oxygen is a competitive inhibitor for Rubisco against the carbon dioxide, and that's why it goes the opposite way. So if they did ask a question on that, it would be an application question. They'd be looking for you to note. Okay, so what else have we got? Our glycerate 3-phosphate. Now, at the moment, the only thing that is needed for the light independent reaction is carbon dioxide. If carbon dioxide levels are low, carboxylase. Alex, to answer your question, so what is the CO? It is carboxylase. So ribulose biphosphate carboxylase, because it is going to fix that carbon into the molecule. So it's a carboxylase. Whereas if you think about respiration, a decarboxylase, DE removal, is going to remove carbon dioxide. So a carboxylase is fixing the carbon dioxide in place. Decarboxylase, releasing it. Guys, thank you so much for the follows. For those of you who are just coming across my profile, welcome. I'm Christine. I go off in tangents all the time. I hope you like what I do. I hope you appreciate, like, share, because it means I can keep doing this, and I love doing this. Anyway, let's get back to the light dependent reaction. Nope, the light independent reaction. And that is the important part. Don't get, for me, right now I'm doing this and I'm going off in tangents because I'm trying to answer questions on TikTok, answer questions on YouTube at the same time, which I love. This is like my happy place. I'm like, ooh, I get to answer questions on whatever you want to know, whilst also teaching you about the subject that I love. However, because I've gone off on a tangent, I have to bring myself back, and my brain is going so quickly right now. For those of you who don't know, I have ADHD, so therefore that is one of the reasons why I'm like, blah, blah, blah. And I have to slow myself back down. I have to double check, and I do that in exams, and I know that I do this when I am excited, I do this when I am stressed, I do this when I am under pressure. I know that about myself, and so therefore I know to slow myself down, I know to double check, and I know to actually correct myself. So what I would say to you is, don't be afraid of making mistakes. And that's easy for me to say because I'm in my 40s and I've done schooling. I mean, I hated school, but I've done the schooling thing. I've done the whole, I'm embarrassed with everyone around me. Oh, everyone's so smart. I'm so stupid. I've been there. I've done that. And I didn't think I would get to where I am today when I was your age. And I wish that I could go back in time and have someone like me, picking myself up, but I wish that I had someone like me who could have explained it in a way that it made sense to me. Now, for some of you, I'm not your cup of tea, and that's completely fine. I wish you the best. That's fine, but I want to help as many people as I can because I held myself back from being able to succeed because I didn't believe that I could, because I didn't get things easily. and so therefore I must be stupid and everyone else is smart, and that's just the way of that life works. That's not true. I just have to work a little bit harder, and if you have to work a little bit harder, you shouldn't have to do it alone. That is what I would say to you, and that's why I do what I do, because I want to help as many of you as I can. So that's why I'm building my resources. That's why I do this. And also, I love what I do. I love teaching. And I don't want to stop that. But I have 2 little girls and I want to be there for them. So that's the reason why I also do what I do. Anyway, back to this. So we have the glycerin. That's like a little advert break from me. Here's a little anecdote of Christine's life. Back to this. We've got our RUBP. We've got our GP, so ribulose biphosphate. We fixed carbon dioxide. It's now lysed into glycerin 3-phosphate, 2 of them. And so the one thing I would say to you is don't forget the light dependent reaction products. Thanks, Alex. This is why I do this, because you guys pop in here and you appreciate that I will go off in tangents. So that ATP is needed. So the ATP that was produced during the light dependent reaction is required for the conversion of our glycerin 3-phosphate, and also we need to reduce the glycerin 3-phosphate into triose phosphate. So we need energy, we need the hydrogen ions and electrons, and then we can do a reduction reaction where we convert the glycerin 3-phosphate into triose phosphate. So what we then need to do is understand, okay, going back, if we don't have enough carbon dioxide, we're going to limit the rate of photosynthesis because we're limiting the light independent reaction. If I don't have enough light, I'm not going to make enough of these products, reduced NADP and ATP. I therefore am going to limit the rate of photosynthesis because I don't have enough to convert my GP into TP. So I need the light dependent reaction and the light independent reaction to be occurring because photosynthesis is both of them together. So I require all these molecules. So if I can't convert GP into TP because there's not enough products from the light dependent reaction, light was my limiting factor on the rate of photosynthesis. Now, if there's no GP available, because there was no carbon dioxide to convert the RUBP into GP, then my reduced NADP cannot drop off its passengers. So if there's not enough of the carbon dioxide, that's going to limit the rate of photosynthesis. My reduced NADP cannot drop off its hydrogens by reducing GP to TP. Therefore, cyclic photophosphorylation can occur, but not the non-cyclic photophosphorylation because there's no oxidized NADP able to pick up the hydrogens and electrons from the light-dependent reaction. Yes, I was about to say, yes, I'm live. No, I am live right now, but no, I'm not going live tomorrow, I'm afraid. I go live on a Monday and a Wednesday. Monday is dependent. Wednesday is the day that I choose to do my actual lives. Every week is a Wednesday at 9 o'clock. But all my lives are recorded on my channel. So if you haven't gone over to my Biology with Christine YouTube channel, the whole course is there. 2 years worth of a course. All of my videos are there for you. Very short and specific to topic areas. And I do go live on a Wednesday. Okay, carrying on. 1-triose phosphate will go off to become another molecule, whereas the others need to actually be converted back to RUBP. Now, it's not just 1 And another one, because to get us to the RUBP, oh, that's amazing. Oh, so someone's just written in the comments that they're doing a degree and currently doing a master's. So they have a degree, they're currently doing a master's in biochemistry, but they're loving this live. Thank you so much. That's really lovely. Do you know what? It's a funny one. When I did my degree, I loved doing my degree. Specifically, my final year of uni was amazing. So I loved that because it was really getting into the nitty-gritty bits of biology. And when I went down to be a teacher, so do my postgrad at Cambridge, I panicked that I was complete imposter syndrome. I knew nothing when it came to science. I didn't know GCSEs from Key Stage 4, Key Stage 5, Key Stage 3. All of that was confusing because I was from Scotland. And then when I came back to do my master's at Cambridge as well, I loved learning again. I loved those bits that I'd forgotten about, that I hadn't done much on. It's just, I've realised over the years that I love to learn. I just didn't believe that I could learn. And so because of that, I basically hindered myself. And one of the things that I actually did my master's on, for those of you who are just coming across my profile, my master's was on your academic emotions linked to learning and your self-concept, your belief in yourself, and how that can have an effect on whether you are able to progress and achieve or not. If you go into an exam and you say to yourself, this is going to be hard, but I'm going to try my best and I will just do everything that I can to show what I know, you are more likely to succeed than if you go into that exam going, well, what's the point? I'm going to fail anyway. So just believe that, yes, it's going to be hard, but if you've done the work, if you've really tried your best, you are going to be able to show that on paper as much as you can. But obviously you have to do the work up to that point. And that's why I do these lives, so that you can come along and actually watch my lives when I do them, or watch them back when you're not here, to be able to go and watch the videos to catch up on bits. So, and you just said you got a test on this and Respiration Friday. Are you and Alex in the same class? He's got his tomorrow. And you're worried because the exam questions are so hard to apply. Well, actually not. Do you know what? Let me show you a question. Let me just pick up a random question, because we've been doing this. I am going to bring a question in. There's a question I absolutely love on this. Give me a second. I did it in my group tuition. Oh, I've got to find it now. It was module 5, photosynthesis. Here we go. I absolutely love this question. So if you give me 1 second, I will bring in. Here we go. And some of you will have seen it, because it's a question that OCR have put out and used in the past, and everybody hates it. However, I absolutely love it. And I made it into, because I'm all about my diagrams, I made it into diagrams. Give me 1 second. Right, hold on. So. I always feel very worried I can't apply your knowledge to the question, but I can for the rest. I got a C at biology A-level, but I find degree level so much easier. Do you know, it's a funny one. I got a B at biology, but it would have been AS level. And then I had a number of years out, and then I went to uni when I was 20. And when I was 20, I actually really did find that I liked it and knew it more than I did when I was at Skill. Oh wow, you're going to be doing a PhD. That's really cool. Okay, here's the question. I love this question, but everybody hates it. Students hate it, but I love it. Okay, so what have we got? You have got here, using the results shown in the table. So this is the table they gave the students in the exam. Explain what can be concluded from each tube or pair of tubes about the light-dependent stage of photosynthesis. So you've been given a table of results, right? So someone has got a table of results. they've done an experiment and they want you to look at tube A and only tube A. So what we're going to look at is see what they've said. Now, this is an interesting one because if you're AQA, you have to do centrifugation, but with OCR, you don't have to do centrifugation, which is really fascinating that you don't actually have to do that for the centrifugation, but you do have to be able to apply the knowledge of centrifugation. So what have they told us? They've told us that there are contents in tube A, tube B, C, D, and E. They've told us that they've boiled it for 5 minutes. They've told us that they've used DC pip. Now, for those of you who don't know, DC pip is an indicator that will accept electrons. So it acts like the NADP and therefore accepts the electrons for the non-cyclic photophosphorylation. So when we look at this, what we've got is, well, here is my sample. And what I have taken from my sample is the supernatant. So that basically means that I have got the stroma. So this is my chloroplast. And what I've got in here is my stroma. And my stroma is this light green color. And if I have boiled it for 5 minutes, no, I haven't, because they've told me I haven't boiled it because it's the liquid. I've got DC pip that's been given to it. Yes, it has. So the DC pip is blue. So when I put DC pip in there, it is blue. If I illuminate it with light, if I put a light source on my sample, what happens to it after 5 minutes? It stays the same color. It stays blue. So what does that tell us using only tube A? Well, we know the light dependent stage of photosynthesis. We would have the light being absorbed by the photosystems, the electrons being excited, the electrons being donated to the electron transport chain, and they will move through to the final electron acceptor. And we know in this case, the DC pip would be the final electron acceptor. And so if we know the DC pip is the final electron acceptor, we therefore need to know that if there's light available and photosystems are in our stroma, then that color would change to be colorless. It doesn't change, it stays blue, so we can conclude that the place where the light is absorbed, the photosystems, are not in the stroma. So that's one of the things that we can use as a way of concluding. So our photosystems are not in the stroma, therefore we can say that the light dependent reaction only occurs in the thylakoid. So that's for tube B. Now, if we look at the next, sorry, that's tube A even. If we look at the next one then, and we want to look at, we've got our tube A, we've had our light, it didn't change color. We know that that happened. Well, what happens in tube B? So this time we have what's known as the resuspended pellet. So this was tube A, we're now on tube B. Look how I've got a different color. I've got a darker color. So now I've got the resuspended pellet. So the resuspended pellet is therefore going to contain my thylakoids. So it's not in the liquid, the supernatant. It's now we're looking inside at the thylakoid. So what we need to understand then is if we're looking here, if I boil it for 5 minutes, yes, I have boiled it for 5 minutes. What does boiling do? Well, if we've boiled it, we've got it 100 degrees C. So therefore, we're going to denature any of the proteins and the molecules that are there. We're going to affect their shape. So if we're affecting their shape, then what we have got is we have got a denaturation of the things that are going to be useful at absorbing the light. So those proteins, those enzymes, the electron transport chain, they are going to be removed. Is that isolating organelles? Yes. How do you know thylakoid is resuspended and stroma is a supernatant? So you have a little bit of information above in the stem of the question that tells you about what the supernatant is and what the resuspended pellet is. Yes. So Darrow, yes, homogenization, filtration, ultrafiltration, that's part of the AQA spec. It's not part of OCRs, but it's good to know that these are things that we're going to use. So Anya, it's not the NADPH, which is the reduced NADP, or the NADP that's going to be denatured. It's the electron transport chain. It's all the proteins that are in the electron transport chain, the ATP synthase. So what you have to understand is NADP is reduced through the use of an enzyme called NADP reductase. So we're going to denature that by boiling this for 5 minutes. So if we boil it for 5 minutes, then we have got the DC-PIP being added and the color doesn't change. Yeah, I know it's 10 o'clock. I'm literally going to finish. Normally I finish now, but we're going to carry on. Now the next one is about C and D. So C, we've got the resuspended pellet. We're going to use DC-PIP. We're not going to shine light on it because it's in the dark and it stays blue. Whereas if we do the same one and we therefore shine some light on it, it changes to be colorless. So we can use C and D as a way of showing light is required for the light dependent stage, and because the light is being absorbed by the photosystems within the thylakoid, that therefore means that the electrons are going through the electron transport chain, and that therefore means that we are going to have the DC-PIP as the final electron acceptor. And that was in the question above. Oh, does it show C isn't light dependent and D is? It's not showing that C isn't light dependent. It's all showing about the light dependent stage. It's a way of you comparing if there's no light, are electrons donated anyway? It's a comparison between when there's light illuminated and when there's light not. Do electrons get donated to DC-PIP without there being light present? No, they don't. Because when the light wasn't present, the DC-PIP did not change color. It stayed as being blue. So there are no electrons being donated within the sample. And so that therefore means that we can compare those two together to say light is required for the electrons to be donated in the light dependent reaction. And the last one then, distilled water. We've got distilled water, we've got no boiling, we've got yes DC-PIP. So we're going to shine a light on it, but it stays blue. So that shows you that DC-PIP does not spontaneously reduce. It's a control for you to compare it to. Right, I am going to stop there because I am getting tired. And these questions are tough. You're absolutely right. But they're there to assess your understanding and assess your way of applying the knowledge that you have got to the context that's in front of you. And that's the thing that students miss out on. They will literally memorize the content and then not be able to apply that information to the random bit of an experiment that they've been given, because they're not thinking about, oh, why is light required? Oh, why is that? So it's about putting it all together, and that's so hard. How do you do this better? The applying bit. The applying bit comes through practice, lots and lots of practice. So lots of practice and looking at the mark schemes and seeing where and how they've given the marks and why. And if you don't understand it, if you don't understand the mark scheme and why that's the answer, then asking questions, put you off because I can't. Don't let it put you off. If you can't, so the first thing I would say, practice using those questions. Don't let it put you off, because every mistake you make is a step of you moving in the right direction to getting it next time. So practice, have a shot. If you get it wrong, you get it wrong. Who cares? Nobody's going to see you're doing it on your own. Number 2, use the mark schemes to figure out what the perfect answer would be. Now, don't just write the mark scheme again. That's a waste of time. Use the mark scheme to turn that into your own answer in your own words. Because if you can't turn the mark scheme into your own words, if you're just rewriting the mark scheme out, you're not learning, you're not thinking about it, you're not considering how you can then use that later on when you get a different type of question. Okay? So it's really important that when you're doing this, that you're finding out ways to make it stick in your head. And using the mark schemes and memorize the mark schemes only work if you get that exact same paper again. Using the key words relating to the topic of the question, that's what's going to help you. Oh, thank you, Anya. Right, I am going to finish, guys. Thank you so much for joining me tonight. I will be back next week at the same time at 9 o'clock. If I can, I will go live on Monday at 9 o'clock. It just depends on whether I have the time or not. Thank you. If you have bought my revision guides, thank you so much. They are still currently at 99p each. And so do check them out. And I am currently working on some more. I'm actually also currently working on some practice papers. I'm trying to make a paper 1, 2, 3, 2 sets of 1, 2, 3s for you. So do keep watching. I will keep doing as much as I can to help you. But I appreciate for those of you who do like, watch, comment, write questions, share these. The more you do that, the more I can reach more people, and I can then obviously help as many as I can. So thank you for all joining me, and have a good evening.